Hello, my dear students. Today we will discuss about microbes in medicine. So generally, algae and fungi, they show the positive role and negative role in the environment. So we discuss positive roles, the microbes in medicines. So wonder drugs, penicillin, streptomycin, chloramphenicol, and tetracycline. These are called wonder drugs. Penicillin, streptomycin, chloramphenicol, and tetracycline. These are the wonder drugs. These all are used as a antibiotics. Streptomycin genus are named with the suffix mycin. So which antibiotics are obtained from the Streptomyces genus? Now they are end with mycin. Whereas those that are derived from the Micromonosporma, these are the bacteria produced antibiotics with suffix or end with mycin. So Streptomyces is fungi. Micromonosporma is a bacteria. The Streptomyces genus producing antibiotics are ended with mycin. These are fungal members, and Micromonosporma. These are the bacterial members. They are also produced the antibiotics ending with mycin. Let us we see some of the antibiotics, beta-lactam antibiotics. The popular beta-lactam antibiotic is penicillins. So this is the first discovered antibiotic by Alexander Fleming, Penicillium notatum. So this Penicillium notatum produced the antibiotic penicillin G, popularly known as penicillin G. The chemical compound is benzyl penicillin. And now they are produced commercially Penicillium chrysogenum, that is called penicillin V, phenoxymethyl penicillin. So Penicillium notatum produced the penicillin G, Penicillium chrysogenum produced the penicillin V. Chemically, Penicillium notatum produced the benzyl penicillin, Penicillium chrysogenum produced the phenoxymethyl penicillin. The first penicillin antibiotic produced from the Penicillium notatum by Alexander Fleming. So most of the penicillins are active against gram-positive bacteria. Why? Because these beta-lactam antibiotics to destroy the cell wall, gram-positive bacteria, the cell wall is made up of murin and peptidoglycan. They immediately destroy the murin or peptidoglycan, so they can easily destroyed by these antibiotic penicillins, are popularly known as beta-lactam antibiotics. The commercially penicillins first purification and production by work of Flory, Chain, and co-workers. So Flory and Chain first produce the penicillin in commercially by the purification and production work. And another type of beta-lactam antibiotics, cephalosporins, they are also act on the cell walls. Best example, some of the cephalosporin producing antibiotics, Cephalosporium ectrimonium, Streptomyces clavagarius, Nocardia lactamundras. Cephalosporium acromonium, Streptomyces clavagrinus, Nocardia lactamundrans. These are produced by cephalosporins. And carbapenems, these antibiotic produced by Streptomyces catali. These carbapenems produced by Streptomyces catali, it is not active against methicillin-resistant Staphylococcus aureus. M means methicillin, R means resistance, S means Staphylococcus, A means aureus. MRSA, it is a superbug bacteria. It is antibiotic resistant superbug bacteria. It is commonly seen in all hospitals. That is MRSA, methicillin resistant Staphylococcus aureus. And another type of antibiotics, tetracyclines. The first tetracycline is aureomycin. Commercially, they are produced from Streptomyces aureofaciensis. So tetracycline, these are also produced from the microbe, which is fungal member Streptomyces aureofacius. The first tetracycline is aureomycin. And quinolones, these are also produced by microbes, Pseudonocardia species of actinomycetes. Quinolones are produced by Pseudonocardia actinomycetes, microbial members. Next, antibiotics are which are obtained from the microbes, aminoglycosides. These are obtained from Streptomyces species, which are fungal members, and Micromonospora species, which are bacterial members. Next one is fatty viracins. These fatty viracins are obtained from Streptomyces microflavus. And most important antibiotics are obtained from the macrolides, popularly group of known as macrolides, are obtained from the fungal and bacterial members. Azithromycin, clarithromycin, rokitamycin. And chloramphenicol, it is obtained from Streptomyces venugilae. Chloramphenicol is obtained from Streptomyces venugilae. Lincomycin is obtained from Streptomyces lincolensis. Lincomycin is obtained from Streptomyces lincolensis. Griseopulvin, it is obtained from Penicillium griseopulvum. This griseopulvin, which is used in treat of athletic foods and some skin disease in human beings. So griseopulvin is obtained from Penicillium griseopulvum. Is treated with athletic foods. And some of the antibiotics and produced organism and activity and site or mode of the action. Cephalosporin, producer organism is Cephalosporium acromonium. Broad spectrum, they destroy the bacterial cell wall. Griseopulvin, Penicillium griseopulvum, dermatophytic fungi, they destroy the microtubules. Bacitracin, obtained from Bacillus subtilis, they destroy the gram-positive bacteria, especially they damage the cell wall. polymyxin B, Bacillus polymixa, act on gram-negative bacteria, destroy the cell membrane. Amphotericin B, Streptomyces nodosus, act on fungi, destroy the cell membrane. Erythromycin, Streptomyces erythraeus, act on gram-positive bacteria. They damage the protein synthesis of the bacteria. Neomycin, Streptomyces fridae, broad spectrum, they act on gram-positive and negative bacteria. They destroy the protein synthesis in the bacteria. Streptomycin, Streptomyces griseus, act on gram-negative bacteria. They destroy the protein synthesis. Tetracycline, Streptomyces rimosus, or Streptomyces aureofaciensis. It is also broad spectrum. They kill the gram-positive and negative bacteria. They destroy the protein synthesis in the bacteria. Vancomycin, Streptomyces orientalis, act on gram-positive bacteria. They destroy the protein synthesis. These are the some of the important antibiotics: cephalosporin, Cephalosporium acromonium, Griseopulvin, Penicillium griseopulvum, Bacitracin, Bacillus subtilis, Polymyxin B, Bacillus polymixa, Amphotericin B, Streptomyces nodosus, Erythromycin, Streptomyces erythraeus, Neomycin, Streptomyces fridae, Streptomycin, Streptomyces griseus, tetracycline, Streptomyces rimosus, or Streptomyces aureofaciensis, Vancomycin, Streptomyces orientalis. And some of the antibiotics produced bacteria, especially Micromonospora species. Anthracycline, it is derived from the genus Micromonospora. Anthracycline used as a drug in cancer treatment. So which drug used in cancer treatment? Anthracycline. And some of the antibiotics produced by Micromonospora, microcins A and B, Micromonospora fuscus, and rosamicin, Microspora rosaria, mutamicins, Micromonospora inuensis. Most important antibiotic, gentamicin. This gentamicin is obtained from the Micromonospora purpurea. It is bacterial member. Gentamicin is obtained from the Micromonospora purpurea. Rifamycin S, Micromonospora rifamycinica. The most important, one of the cancer drug which is obtained from the Micromonospora, anthracycline. Gentamicin is also obtained from the Micromonospora purpurea. And microbes in agriculture. So microbes in agriculture, mycorrhiza. Mycorrhiza is popularly known as arbuscular mycorrhizal fungi, AM fungi. Arbuscular mycorrhizal fungi. Previously it is known as VAM, vascular arbuscular mycorrhizal. So VAM nowadays called AM fungi. AM fungi means arbuscular mycorrhizal fungi. Previously it is known as VAM, vascular, arbuscular mycorrhiza. This mycorrhiza belongs to glomerulus order and class Zygomycetes. So mycorrhiza belongs to which class? Zygomycetes order glomerulus. The meaning of mycorrhiza, the symbiotic association between the fungus and higher plant roots. The symbiotic means both organisms are getting benefit. They are called symbiotic association between the fungus and higher plant roots. Different type of mycorrhizas are present. The popular mycorrhiza are arbuscular mycorrhiza, generally belongs to glomeromycota. Arbuscular mycorrhiza belongs to glomeromycota. Ectomycorrhiza, these species belongs to Basidiomycota or Ascomycota. So ectomycorrhiza popularly belongs to which fungi? Basidiomycota or Ascomycota. Arbuscular mycorrhiza, popularly known as endomycorrhiza. Endomycorrhiza belongs to glomeromycota. So endomycorrhiza known as arbuscular mycorrhiza. Second one is ectomycorrhiza. Ectomycorrhiza belongs to Basidiomycota and Ascomycota in the fungal members. Endomycorrhiza or arbuscular mycorrhiza belongs to glomerulomycota. Generally, they absorb the phosphates from the soil. And another important microbes used in agriculture, rhizobia. The role of 6 genera of Rhizobiaceae bacterial family in agriculture production has also been well documented, with many cases of yield increase which inoculated. So generally they are known as rhizobium singularly, or plurally it is called rhizobia. Singularly it is rhizobium. So the first known rhizobia is Rhizobium leguminosarum. Rhizobium leguminosarum. Now it is popularly known as Mesorhizobium. Rhizobium leguminosarum is known as Mesorhizobium. These are used in agriculture to fix the nitrogen. Atmospheric nitrogen is converted into ammonia or ammonium and fixed in the soil. It is called nitrogen fixation. Many of the microbes which belongs to Rhizobiaceae family to fix the atmosphere nitrogen in symbiotic manner or free living manner. So in the symbiotic manner, some of the rhizobium species are to fix the nitrogen. One is Brody Rhizobium japonicum. They fix the nitrogen in which plants? Glycine max. That means Brody Rhizobium japonicum to form the root nodules only in glycine max plants. So they call host specificity. So symbiotic association and host specificity is seen in a symbiotic nitrogen fixing bacteria. Generally, they are present in the root nodules. So when we discuss the Fabaceae family, we give the detailed account how their root nodules are formed and the nitrogen fixation lesson is also with detail elaborated. Just we saw this chart, Brody Rhizobium japonicum and glycine max, Rhizobium predi, glycine max, Rhizobium fascioli, Fasciolis vulgaris common bean, Rhizobium melioti, Medicago sativa or alpha alpha, Cowpea rhizobia or Rhizobium species, Vinga species, Cowpea, and Rhizobium trifolium species, clovers, Rhizobium leguminosarum or Mesorhizobium Rhizome sativum, and Rachis hypogea. These microbes, especially Rhizobiaceae family, to form the root nodules in Fabaceae family and fix the nitrogen in symbiotic manner. So they are useful in agriculture. And other nitrogen fixing symbiotic bacteria except Rhizobia. So some of the actinomycetes like Frankia also to fix the nitrogen in symbiotic bacteria, and other symbiotic bacteria are Azotobacter, Azospirillum, Acetobacter, Azoacrus, Burkholendria, Herbaspirillum. These are also symbiotic nitrogen fixing bacteria. So other than Rhizobium, popularly Frankia, actinomycetes to fix the nitrogen in symbiotic manner. Along with Frankia, Azotobacter, Azospirillum, Acetobacter, Azoacrus, Brucholendria, and Herbaspirillum also to fix the nitrogen in symbiotic manner. And other plant growth promoting bacteria, plant growth promoting bacteria, PGPB, Pseudomonas bacteria, Pseudomonas fluorescensis, Pseudomonas putida, Pseudomonas gladioli. These Pseudomonas are also plant growth promoting bacteria. And some of the bacilli bacteria are also Bacillus subtilis, Bacillus cereus, Bacillus circulensis. And some of the other bacteria, Flavobacterium species are also plant growth promoting bacteria. So Pseudomonas, Bacillus, Flavobacterium are other plant growth promoting bacteria. Agrobacterium radiobacter used to control the crown gall on several plant families. So crown gall disease reduced by the Agrobacterium radiobacter. So this Agrobacterium radiobacter used in agriculture to control the crown gall disease. Bacillus subtilis to suppress the Rhizonatia solani infections. Cereal rot, root rot diseases caused by Rhizoctonia solana. So these are controlled by bacteria Bacillus subtilis. So mostly Bacillus bacteria are used to increase in the crop yielding. So they are called YIB, yield improving bacteria. So they suppress the different type of root rot diseases caused by Rhizoctonia solani, which is controlled by Bacillus subtilis, popularly known as YIB, yield improving bacteria. And biocontrol fungi, various non-pathogenic saprophytic strains like Rhizoctonia, Fusarium, Trichoderma species are used to reduce the damage of root rots, wilts, damping up, and bare patches caused by the pathogenic fungi. So some of the fungals are used to control the different type of root rot diseases, wilt diseases, damping of diseases by Rhizoctonia, Fusarium, Trichoderma species. And microbes in environment, bacteria and fungi are able to biodegrade or detoxifying substances through various ways. Thereby, microbial processes are extensively used for bioremedication. So, the large compound molecules are split into simple degradable compound molecules, which is used by microorganisms like bacteria and fungi. They are called bioremediation. So which fungal members are used in bioremediation? They are called mycoremediation. Which of the fungal members are used in bioremediation? They are called mycoremediation. And biostimulation and bioaugmentation process promotes the rate of degradation of organic and inorganic pollutants. So biostimulation and bioaugmentation is process promote the rate of degradation of organic and inorganic pollutants. So in the biostimulation and bioaugmentation also, some of the microbes are used. So Pseudomonas, Spingomonas, and Mycobacterium are involved in biostimulation and bioaugmentation. Pseudomonas, Spingomonas, and Mycobacterium. Popularly, they are used in oil degradation.